Alejandro. I am a postdoctoral researcher at the University of Canberra, and I've been working with the application of eDNA in biosecurity for a while now. eDNA is quite simply DNA that we shed into the environment. It's DNA that falls out with our skin, with our waste products, and as we also shed DNA, so do animals in the water, in the soil, even in the air. So we can collect that DNA that is shed by these organisms and test it for their presence. For a long time, we've been relying on quite a bit of heavy machinery, on huge machines and thermocyclers that we need to use in the lab. But that has changed for almost 4 years now. Technology is getting smaller and smaller, which is the case of this little machine here. That is called a MinION sequencer. It's a pocket-sized machine that allows you to sequence all the DNA that is in a given sample that you've collected from the environment. We essentially take DNA that we can collect from the water, for example, and we throw it into this little machine, and it's going to sequence the genetic code of any DNA that is in that sample. With that in mind, this machine allows us to quite efficiently and with a little bit of time know exactly what animals are in a given sample. So we can use it in, let's say, in lakes to make sure that we don't have any invasive species or to detect invasive species. We can use it in biosecurity to understand what species are coming in at border control. For example, with aquarium fish, when you would get fish from overseas, there's some species that are not legal for import to Australia. We can use this technology to find those non-permitted species. The same goes for samples in the environment, where we can detect, for example, cane toads in different ecosystems where people would normally not be able to go. And it just makes everything faster, simpler to work in science. So with this, we can just take the lab outside and test all of these samples and this technology in a different environment.